In this class, we are going to discuss about the mechanism of opening and closing of stomata. As you are aware that stomata are minute pores present on the leaf surface, and these pores are mainly useful for the evolution of water vapor during the process of transpiration, and also first one is evolution of water vapor, and also these pores are useful for exchange of gases like O2 and oxygen and carbon dioxide. And these pores are normally present on both the surfaces of the leaf. That is, they occur on upper epidermis and lower epidermis. In most of the dicots, the leaves are described as dorsiventral leaves. In this case, the stomata occur on both the surfaces of the leaf, that is on the upper surface and also lower surface, but the number of stomata are more on lower surface when compared with the upper surface. So in dorsiventral leaves, number of stomata are more on the lower epidermis, when compared with the upper epidermis. And in the case of monocots, if you observe the monocots, the leaves are described as isobilateral leaves, where the stomata are present on both the surfaces of the leaf, and they are equally distributed. Their number is equal on both the surfaces. In the case of dorsiventral leaf, the stomata occur on both the surfaces, but the number of stomata are more on the lower surface when compared with the upper surface. That is the condition mostly observed in dicots, and such leaf is called as a dorsiventral leaf. But in the case of monocots, stomata occur on both the surfaces of the leaf, but the number of stomata are equal on both the surfaces, and such type of leaf is known as isobilateral leaf. And these stomata are normally, they are minute pores, and each pore is, suppose if we observe a stomata, each pore is guarded by means of two guard cells. These are the guard cells, and these guard cells are in turn, they are surrounded by means of these cells, which are called as subsidiary cells. This is GC, and this is the stoma. So the entire apparatus, the entire structure is called as the stomatal apparatus. So this is the actual pore, and this pore is guarded on either side by means of two kidney-shaped or bean-shaped cells, and these two kidney-shaped cells are in turn surrounded by subsidiary cells, and this entire structure is known as the stomatal apparatus. And if we observe the guard cells, these guard cells are mainly, these are the guard cells, and these guard cells are mainly responsible for the opening and closing of stomata. So the opening and closing of stomata mainly depends upon the turgidity of the guard cells. Turgidity means when the guard cells are filled with water, they become swollen and the stoma opens. That is called as turgidity. In dicots or in most of the plants, the stomata open during daytime. So during daytime, these guard cells they become turgid with the absorption of water, and in that turgid condition they will open. And if you observe the wall of the guard cells, this is the wall of the guard cell, and the wall of the guard cell is not uniform. The wall that is adjacent to the pore, this is the wall adjacent to the pore. So this wall is thick. So the wall adjacent to the pore is thick. This is thick wall. The wall away from the pore is thin. This is thin wall. This is thin wall. This is thick wall. So that is the morphology of the guard cell. So the wall around the guard cell is not uniform. The wall adjacent to the pore is thick walled and it is elastic. Thick walled and elastic. The wall away from the opening is thin walled and also elastic. So in addition to this, if we observe the guard cells, we can also observe some of these microfibrils. These are the microfibrils. And these microfibrils are arranged radially instead of longitudinally. They are arranged, microfibrils are arranged radially. So the radial arrangement of microfibrils also is useful for stretching the guard cell. Suppose when the guard cells, when the stomata are in closed condition, both the guard cells are expressed like this. So this is the closed condition. This is the closed condition of the guard cell, and this is the opening. This is the opening condition of the guard cell. This is the opening. So here, this is the stroma. The stroma is closed here. So whenever water enters into the guard cells, the guard cells becomes swollen, and as a result of this swollen condition, the thick wall of the guard cell that is adjacent to the pore is stretched. It becomes stretched like this. It becomes stretched, and because of this stretching, the thin wall of the guard cell also it becomes swollen and it is enlarged, and the stroma opens. So whenever the stroma opens, the water vapor will come out. In the evening conditions, whenever the guard cells lose water, they become flaccid. And in this flaccid condition, the wall of the guard cells adjacent to the stroma, they become oppressed and the stroma becomes closed. That is how the opening and the closing of stomata depends upon the rigidity and plasticity of guard cells. In order to explain the mechanism, how the guard cells becomes rigid and how they become plastic, so many theories have been proposed by different scientists. Of all those theories, the theory proposed by Levitt is accepted by most of the scientists. And this Levitt's theory, it was proposed in the year 1974. This theory is known as potassium pump theory. That theory is called potassium pump theory. According to this potassium pump theory, in photoactive stomata, due to the accumulation of, suppose if we observe the guard cells, so there are two types of stomata we can come across. The first one is photoactive stomata. The photoactive stomata means the stomata which open during or in presence of light. Such stomata are called as photoactive stomata. Then in presence of light, what will happen? In presence of light, if we observe the guard cell, there is influx of potassium cations. There is, due to the influx of potassium cations, there is a gradual accumulation of potassium cations inside the guard cells in presence of light. Simultaneously, there is this is called influx. There is efflux of H plus. So in presence of light, two reactions or two things will occur. The first one is there is a gradual entry of potassium cations from the subsidiary cells into the guard cells. Entry of potassium cations into the guard cells is called efflux, and this is done only in presence of light. Simultaneously, that is at the same time, hydrogen protons present in the guard cells, they send outside, and this sending of hydrogen protons to outside or expulsion of hydrogen protons to outside is called as efflux, and this is an active process. This is an active process, for which, suppose if we observe the entry of potassium cations, where we can represent the line as dotted, that is passive. This is the passive. So influx of potassium cations is passive. There is no need of any energy for the entry of potassium cations. That can be represented by this dotted line. But the expulsion of hydrogen protons from the guard cells, it requires energy. So it is an active process. Hence, it can be represented by means of this thick line. So, because of this accumulation of these potassium cations, there is a gradual increase. In the pH of guard cell increases. Because of this type of reaction. And as the pH of guard cell increases, there is also a simultaneous entry of chloride ions from outside. At the same time, chloride anions also enter the guard cells. So by the entry of these guard cells, due to the guard cells becomes electrically equilibrium, at this condition, the water potential of guard cells decreases. Here, two types of things will occur. First one is due to the accumulation of potassium cations, the pH of the guard cell increases. At the same time, chloride anions also enter into the guard cells passively. Because of the entry of these chloride anions, gradually the water potential of the guard cells decreases. As a result of this decrease in the water potential, water from the subsidiary cells enters into the guard cells, and the guard cells, as a result of this endosmosis, they become swollen. So water always moves from a place of higher water potential to the region of lower water potential. Now the guard cells where the water potential is decreased, now water from the subsidiary cells or from the region of higher water potential, water slowly enters into the guard cells, and the guard cells becomes turgid, and the stroma, that results in the opening of the stroma, opening of stomach, and this will occur during daytime. In the evening times, if we observe the guard cell, some reactions or some changes will occur inside the guard cells. So in the evening, due to water stress conditions, a hormone called abscisic acid, ABA. Whenever the plant experiences water stress condition, అంటే నీరు లేని పరిస్థితి, ఈవినింగ్ టైమ్స్ లో జరుగుతుంది, in the absence of sunlight, a hormone called abscisic acid is produced inside the guard cells. And this abscisic acid, it acts as a natural, it is a natural anti-transplant. It occurs in the plants, and in presence of this ABA, gradually the water potential of the guard cells increases. Some changes will occur due to the presence of this ABA. The first change is the water potential value inside the guard cell increases, and the second change is the pH of the guard cell decreases. Gradually, water from the guard cells, suppose if this is the guard cell, water from the guard cells, gradually what will happen in this condition? The potassium cations which are present inside the guard cells, they are expelled out. Guard cells, stomata open అవ్వాలంటే potassium cations guard cells లో enter అవ్వాలి. But in the evening times, the potassium cations will expel out. At the same time, chloride ions also they expel out. The hydrogen protons make their entry. Hydrogen protons slowly enter into the guard cells. With these changes, the pH of the guard cell decreases. The water potential of the guard cell increases. In this condition, the water from the guard cells, it will be expelled out. This process is known as exosmosis. In the daytime, water will enter into the guard cells. This process is known as endosmosis. In the evening times, water will come out of the guard cells by a process called exosmosis. As a result of this, the guard cell becomes flaccid. And slowly the stomata closes. So it leads to the closure of the stomata. So closure of the stomata lo ee reactions, opening of the stomata lo ee reactions jarugutayi. That is how the opening and closing of the stomata depends upon the turgidity and flaccidity of the guard cells. So this type of mechanism, it was given by Levitt. This mechanism is known as potassium pump theory. Only the potassium cations are mainly responsible for opening and closing of the stomata. And all this mechanism is applicable to photoactive stomata. Photoactive stomata means those stomata which open in presence of sunlight. And this is a common feature. We can observe this feature in most of the angiosperms. But there are some plants of second one, scotoactive stomata. These stomata are found in cacti and also in plants like bryophyllum, where the stomata open during night times. Then what is the mechanism? Normally stomata open during daytime in presence of sunlight, but in the case of these plants like cacti and bryophyllum, the stomata will open during night time. Then our problem is, what is the actual mechanism that is involved in the opening of the instrumenta during night times? According to the mechanism, during night times there is a water potential gradient. A water potential gradient is mainly developed in God cells, due to the presence of or due to the formation of some organic acids, like malic acid. In these plants like cacti and bryophyllum, an organic acid called malic acid is produced during daytime. As a result of this malic acid, due to the accumulation of this organic acid called malic acid, there is a water potential gradient is established inside the guard cells. Due to the establishment of this water potential gradient, slowly water enters into the guard cells. So during night time, the guard cells they become turgid and the stomata opens during night time. So this is the mechanism that is meant for the opening of stomata in the case of these plants like cacti and bryophyllum. Now the next question is the significance of transpiration. As you know, transpiration means expulsion of water in the form of water vapor from the aerial organs of the plant. So by this transpiration, 99% of water is lost from the plants. Only 1% of water is utilized by the plant for its metabolic activities. So then we can infer. So this transpiration is not at all useful for the plants. It has an evil effect. But there are so many benefits also by this transpiration that comes under this significance. The one is there is absorption. The first one is absorption and transportation of water is possible by this process called transpiration. And the second one is it is also useful for photosynthesis. And it is also useful for the transport of or mineral transport to various organs of the plant body. And because of this transpiration, there is 10 to 15 degrees centigrade reduction of temperature in plant body. Due to this this is known as evaporization, and water is evaporated from the plants by this transpiration. There is a cooling effect to the plant. This cooling effect by this 10 to 15 degrees centigrade of temperature is reduced in the plant, that will give a cooling effect, and that evaporization is mostly useful for the plant body to maintain its temperature also. And the next important useful the significance of transpiration is it keeps the plant cells in turgid condition. So plant cells in a turgid condition float, and this turgid condition of the plant cells is responsible for maintaining the healthy condition of the entire plant body, and plant parts also shows movement with the help of this turgidity. These are the 5 important significant features by this transpiration. And the transpiration, even though there are so many benefits, but as we know, because of this process, 99% of water is lost. In spite of this loss, plants can afford to perform this transpiration. Hence, so there are benefits, there are also harmful effects by this transpiration. So Curtis, a scientist by name called Curtis, he described it as transpiration is a necessary evil. డే స్టూడెంట్స్ చిన్న ఇన్స్ట్రక్షన్ ఏంటంటే మీరు ఈ లెసన్ వీడియోలో చూస్తున్నప్పుడు మీరు దాన్ని స్క్రీన్ షాట్ తీసుకొని ఇందులో మేము ఏంటంటే ఇన్ఫర్మేషన్ మీ బోర్డు మీద రాస్తున్నామో అదంతా కూడా యూ జస్ట్ నోట్ డౌన్ ఇన్ నోట్స్ ఇన్ యువర్ బాటనీ నోట్స్. సెకండ్ ఇయర్ బాటనీ నోట్స్ పెడతారు కదా, అలాగే నేను నోట్స్ పెట్టుకొని పాయింట్స్ అన్ని కూడా రాసి నేను మళ్ళీ నెక్స్ట్ వెనెవర్ మీట్ అగైన్ మన క్లాస్లో మనం కలిసేటప్పుడు ఆ నోట్స్ నేను చూస్తాను. ప్లీజ్ మేక్ ఇట్ ఏ పాయింట్.